Yeah, so machine learning and deep learning methods I already discuss what is ensemble method. And okay, so what is there in the microRNA deep learning and image deep learning will cover, and also will cover some examples on coronavirus and data analytics in actual programming in Python. So let me introduce you. As I told you that there is a data which is which on which users in machine learning language you select it actually. So you have to know what there are in large number of data existing on the planet, large number of data is existing in the internet, large number of YouTube data is existing in the planet. What you watch, you need to select, and same way what you want to learn to analyze, you need to select. And then you do the on target data, you do the processing. Means you may get a data and you may get suppose this is age versus patient data. You may get data patient when the patient has filled it. They said this, sometimes they had filled date of birth, sometimes they have filled 78, sometimes they have filled 2701 1950. So some other type of value. So you need to pre-process, make them uniform, make the missing data, remove the missing data, remove the data which do not make sense. So in the patient data you asked, patient has put his name, his mother's name. So date of birth is not there. So that data will not be useful for you. So you need to transform it, pre-process it, arrange it in a way that machine can learn from it. Because you can't say that मेरे घर पे आ जाओ और सब कुछ available होगा. You have to prepare. Suppose you're inviting someone for lunch, you have to prepare yourself for that, and that's it. That's what. So machine will be only working with the data which is pre-processed and transformed in a way that machine will learn. So again, I can correlate with the patient data. If the machine you have to provide the gender of the machine. You can say 0 for female, 1 for male, or 1 for male, 0 for male, 1 for female, something like that. So the machine will know have a binary information. Okay. So based on that, when you have transformed data, you can have a model, and model will be then used for interpretation and evaluation. And if you find some problem, so you actually go back to selection process again. And if you finally, you can actually go in this loop several rounds. So those who are training with us and those who are coming and doing with us, they do this, and most of us will do the evaluation. So we'll give you an evaluation concept, how we choose do the evaluation, and so that we'll have an end have a thorough idea about the topic. And once you are doing, you are getting used to it. So those who want to start may contact, and if who have some background and want to train, they can contact after today's class if you are interested. So what is the difference between machine learning and deep learning? So basically, artificial intelligence is so artificial intelligence is yes, artificial intelligence is a major area where you have robotics and all other kind of artificial intelligence and intelligence methods you see. Maybe you have seen in movies also, maybe you have seen in real life also, that sometime your light is switching on and off, what the criteria you have put, and robot is moving things, and all drones and all are coming in large spectrum of artificial intelligence. A sub part of it is machine learning, and then the sub part is neural network, and a small area of neural network is deep learning. And this is how this broad classification is existing, and with few minor changes. And if you have a data set, you can choose based on what you want to use. So recent year, machine learning used in last 20 years in very extensively in bioinformatics and biology. Slowly, people are using more and more deep learning, because in deep learning, uh, processing and other criteria, a machine will take care take care of how data has to be, how data it requires, and it will process the information out of it. Uh, uh, not in machine learning, you have to extensively characterize your data pre-hand. So in biology, there are several applications, as I told you, in genomics, in gene finding, splice prediction, motif identification, RNA, DNA, microarray, systems biology, signaling network, metabolism, again, proteomics, proteinic-based diagnostics, structure prediction, structure function. Then the text mining, such as gene annotation, protein annotation, and also in comparative genomics and evolution, everywhere machine learning and deep learning is used. And by that, when we are saying machine learning, as you saw, that machine learning is covering deep learning also, but deep learning is slowly becoming its own application is so high that it is getting used to it is getting its own sub area is growing very fast and very rapidly. People are using it as an independent unit, and so this is where we are also working, and some of the methods we will be showing you today. Yeah. So what is the need? What is the need? And I probably have shown you that biological data is very heterologous. Like we have DNA data, RNA data, histone-based data, expression data, transcription data, base calling data, pathogenic variant data. A lot of data is there. DNA methylation is there. Tumor data is there. Now the genomic data is rapidly, all this omics data is rapidly increasing. And from heterologous data, you want to extract the information. Then what happens? Then you need tools to predict from such complex data, right? So that's where the machine learning and deep learning can be successful in the uh, process. And in deep learning is just inspired from brain, and therefore it's called neural network. And because there is input, you give input, then there are a lot of hidden layers, like in brain has neurons, and then in hidden layers it will be pre-processed, and output will be given based on the and what is going. So it's sometimes called black box also, the hidden layer, because what is going exactly inside the data layers are sometimes hidden, or many times it is hidden. So same as a brain is signaling or processing its information. So you get it by your senses, and then it processes. Similar way, you give the data, it will be hidden layer will be using, and that's its overall deep learning. And then there are a lot of deep learning based methods in genomics, and recently a paper he has covered, and by Zhang et al. In Nature Genetics, and there they explain how it is. So suppose you have a sequence data, you label that some features like 111 and other kind of signatures 000, and then use it for the training. And then others, one more set of the data you will use at the 0 and 1 as a validation data, and then divide and then a test data. So basically you label your data in a way that a curated data is divided into 3 different categories: training data, validation data, and test data. And then you apply one of the deep learning method or machine learning method, method, and like CNN or RNN. And once you have the method applied and trained, you will evaluate the method using something called confusion matrix, which computes how many true positives are there. It means actual hits. How many false positives are there, and how many false negative, and how many false negative means something which is true but came out negative, uh, and something which is true negative is actually negative. And so this classification is there, and this evaluate based on that, and then you call precision, and also recall based on the four values you have. The different features can be combined, and once you have it, will get you will get the you will make an interpretation and evaluate your results. Okay, so then Perry Young is one of the leading machine learning and deep learning expert in bioinformatics. His group has recently published a paper. What this paper is telling, obviously they are talking about ensemble method of learning, but what they are saying that hope we review this article will bring together a broader audience of machine learning researchers, bioinformaticians, and biologists to foster future research and development in ensemble-based deep learning, and inspire novel bioinformatics applications that are unattainable by traditional methods. So that means we have to come as a community together, including everyone who are doing machine learning, who are doing just genomics and bioinformatics, and those who are not doing first three things, biologists who are doing experiments. So all of them are important. And in this review, there are they have come up explain the methods. So as I explained you what is bioinformatics, you already know what is deep learning, artificial intelligence. I explained you, but how it is very quickly merging and becoming one. And so obviously you know that the artificial intelligence is a broad area, bioinformatics is a broad area, and then in machine learning is sub area in artificial intelligence, deep learning, and then ensemble based learning. And then at the very center, whatever you will jointly see is the ensemble based learning, and that's what they have explained in this paper. And then what are a type of ensemble based learning? You have a particular first method is called begging. Begging means you have data which is perturbated, and you have a classifiers. You make a classifier and go for a voting function. So basically, you go with like a democratic way. Okay, you go data and classifier. You vote each of them, and then one function. That subgroup of data which is voted highest will be considered further, and rest will be not. So that's called begging method. The boosting method, in which the classifier and having having class residues, and then base classifier, you combine both of them and do weighted voting. Weighted voting is that if I'm voting a candidate, I'm not seeing the educational qualification, right? So that is normal voting. But suppose say that to become education minister in India, you need a PhD. Then it is called weighted voting, because you want to wait on something else. At the moment, no ministry in India, or no let people vote for whom they vote. So it's not nothing to do with their education, religion, or whatever weighting function. So anybody can go and win. So if there is alternative voting system where, say that education ministry needs a PhD in education, finance ministry need MSc in or MBA, MSc in finance, something like this, then it will become weighted voting, because only those who have this as weightage will be able to participate. And then in data perturbation method, you even go with the consensus. You derive on a consensus rather than voting. You derive into consensus, and take consensus classification result at the end. And then deep learning in supervised and unsupervised method. Supervised, you understand that some supervised is something when I am doing my PhD, I'm supervised by someone. But suppose I am learning something myself and then I am unsupervised because nobody is telling me what to do. I am learning from my experience or my randomly. Maybe I just watch YouTube and I'm learning something. Then it's unsupervised because I'm not getting supervision. And both method works. Supervised will work when you really know the rules. What kind of rule one has to use. But in unsupervised, there is no rule used because there is no supervision. And then there are different classification and different ensemble methods are used in between. And these are explained in this paper in detail if you want to go. And based on the requirement, people will use different methods, and they have beautifully explained and given several examples. In the different area of biology or bioinformatics, the supervised and unsupervised learning plus deep learning is used. And we see that in sequence analysis, most of the supervised learning is used, okay. So supervised learning is used and the references are available on the paper. Okay, it's moving. Okay, so we see that this in the first sequence analysis, in genome analysis also, supervised learning is used. And what we see in expression analysis, both supervised and unsupervised learning is used. Structural biology or structural bioinformatics, proteomics, supervised is again more used. Systems biology, again supervised is more used. And supervised is predominantly used more in number, and multi-omics, both supervised and unsupervised is used. In image analysis, again, bioimage analysis, again, supervised learning is primarily used. But in general, supervised, you want to learn something, you always go for, it's a human tendency or nature to go with the supervision-based method, and then so do in applications also. Unsupervised learning is used, but it is difficult to use at times. So therefore, one in machine learning, one will go for supervised learning. And so in one of our projects, we have done the microRNA deep learning based method for microRNA target prediction. I will show you what we have done. So recently we have published a paper where we have described what is microRNA and what are the microRNA binding target prediction tools are available. So what we see here is that we have microRNA, pre-microRNA is found in the nucleus, and from nucleus it will come to cytoplasm and function if using microRNA. And microRNAs are actually just 19 to 25 base pair long non-coding RNA, and they will come out from the mature messenger RNA and become independent entity and regulate the cellular processes like cell proliferation, differentiation, angiogenesis, apoptosis. And then there are different tools over from 2014 to 20, almost 15 years, and there are 15 different tools which are using different machine learning and similar support vector machine based tools are developed, and these are listed in the right side. And then we wanted to review the method so that we can use do we need a new method we can cover. And so these are the tools. So as you see that these are the 14 tools in many years. These are specialized from whatever species. And so the MS is the multi-species, and then what kind of feature-based algorithms are used, and they are explained. And then approaches where they have given where we have given that many of them use machine learning and many of them use support vector machine and other algorithms such as base in this case here. And at different point people have used different methods, and we wanted to see how we can use but nobody was using deep learning, as you see, until then. So that was a good idea that we should start machine learning based algorithm using deep learning. So during the process, we also had written a review article on machine learning based microRNA discovery and target prediction. And there we have identified the methods which are based on these are different prediction methods. These are different target prediction methods. First is the microRNA prediction method. Second was the microRNA target prediction. So there is a difference. Yeah. So first one is to predict microRNA itself. Second one, where microRNA will bind in human genome or whichever genome you're looking in. So that is called target prediction. And then third set of the tools are what are the functional implication of such microRNA and microRNA target prediction. So this is third set of tool called functional annotation. Okay, so this is ML and DL in microRNA target prediction. So how features are being selected? The features are being selected using the features are being selected using microRNA structure, microRNA microbinding structure, and the sequence feature. There can be sequence feature, structure feature, thermodynamic feature, and alignment feature. Alignment is this how these are aligning or microRNA to mRNA. And these are the different set of the features. In our study, we have identified 95 such features. So we have so we can identify different data sets, and then what we do normally in this process that we will have different classifier 1 and B and see what is the hyper plane and linear order, and then try to predict from the algorithms what could be the hyperparameters and which tool will fit in optimized parameters. So there are different algorithms, and they are derived from Keras or scikit-learn. These are tools for using machine learning, so existing tools, and scikit-learn and sklearn. And so in our study, we have used optimized the method, and then we wanted to why wanted to predict. What we wanted to do is that we wanted to predict target in which microRNA is complementarily binding, and also we wanted to overcome. So if you do the experimentally, it is time consuming and it's extremely difficult to locate the binding site. So to do so, one needed computational methods, and computational must be faster, especially. And there are, as I told you, there are classical methods which have high false negative, and such as these ones. And then it is only sequence based, so leads into low sensitivity. And then artificial machine learning based methods are there, and they come up with certain imbalance of the data, and then they are without they have some limitations. So we wanted to have a robust method to find a accurate microRNA target prediction using deep learning approach, and we wanted to test it. So what we did was we took the validated we had built a training data set and the testing data set, and we have validated microRNA and pre-microRNA were extracted. A Vienna package was used for generation of positive and negative data set, train machine learning using scikit-learn, and feature selection by lasso and cross validation by scikit-learn again, and then optimize the classifier for microRNA binding. This is training. Training for testing phase. We have new micro pre-RNA duplex was computed using Vienna package. After applying a score of microRNA duplex structure where which was used in a training data set, which same data set was used here. Processing of the score was done, and then we generated a predicted microRNA sequences features. And then when we were preparing our training data set, so we had to prepare a positive data set and negative data set. So we take a positive data set by or positive transcripts we call by just taking transcript from human transcript and mirror-based microRNA, and then look for interaction using TARP based Miranda, and then identify feature using Java based descriptor, and then pre-process it, and pre-process and potentially positive interaction, and that becomes positive data set. Again, we take the microRNA from ensemble. This time what we do is that we mutate all the ensemble data microRNA from by 95% of random mutation. Okay, 95% of random mutation will make so basically almost 9.9 out of 10 base pairs were modified. So the chance of microRNA being there get lower. And then we will do the same way, predict the different interaction using Miranda, TARP, Vienna, TAR base, and look for the feature identification, do the pre-processing, and the first positive one, whatever bad interaction we found, we put it in negative data set, and then we remove the when we have 95% identified, we will still get some good interactions. So those good interactions are again removed, so that we have totally negative data sets, and then these data sets will be used for feature selection and further on training purpose, and using our algorithm using deep feed forward for both positive and data set was created. So data set was positive data set, we took it from database, but negative we have to generate it for our how you can imagine this data set. Suppose you wanted to find patient information. From patient information, you want to think if the patient will get the cancer. So what will be the negative data set for against patient, healthy individual? Okay, so the same way wherever the potential microRNA binding site was there, we have mutated it and then removed, even if still some are left in. So using this analysis we devised it. It took us long time to discuss and prepare this data set actually. And then we used this kind of layers, input layers, hidden layers 2 times, and then output layer, and that's what's called deep feed forward. So you feed it, it will go back and go forward, feed forward. So it will go back and forth in the hidden layers, and then reach to the final. Okay. So then how the data set will look like? Data set will look like we identified when we use this data set, we put lasso for data 95 features feature selection and then training, and we end up with 19 data set only, and we'll show you in detail. But how data will look like: the microRNA corresponding mRNA, if there is seed information, total matches, mismatches, what is the energy. So this is kind of an Excel sheet, will get it, or a sheet, or whatever name you want to give. If you want to see in Excel, you can see in Excel also. So what one is doing, that one is using different feature selection method, and we saw that lasso may predicted 19 different features, and other algorithms predicted less than 10 or 11, and one of them has predicted 17 features. So very high features also will create a problem, and very low features will also create a problem. So lasso is optimal because it is neither very low nor very high. So therefore, we use lasso feature selected by lasso method, and these are the different features: energy, 3' region, microRNA, total GC length, alignment. It's a mixture of different type of and the embedded score is came from lasso, is shown to in the graph here, and the lasso has the different function that is used in 8 layers, and that is shown here. And there are 9 layers in total we identified, and these are the each layer given there, and their weight and total number of batches. And then we compared the sensitivity of our method with the sensitivity of other methods. What we saw here is that the sensitivity is for our method is, this one is overall performing very almost equally good, and then the other methods have lower, or some lower and some higher. So therefore, in this method, we are sure we are very close to 1. And then when you look on the curve, is accuracy under curve, accuracy, true positive, true negative, and other scores are also good for the algorithm which we have after 10-fold cross validation. And so using this, we identified that our prediction is working, and we compared with the other existing method. And what we saw that our method is performing relatively well, because in other methods either one criteria is good or other criteria is bad. So based on that, we can say that this tool was working, deep learning was working well. And then if deep learning was really working well, we wanted to see whether we can apply this to cancer and other data set and see if really working good. So what we saw that there is a pan-cancer data set derived from pan-cancer genomic projects. And these data sets are actually available to public. And then what we did, these are the top top genes found in this study. So what we did was we took our data set, merged with the pan-cancer driver mutations, and wanted to check how many of their driver mutations have this microRNA binding site or microRNA binding potential. And we used different algorithm along with our algorithm. And what we could see that our algorithm is in the last. You see that most of the algorithm is having red, 0, no interaction. In our algorithm, we see that most of gene have some interaction, and so positive score is there. And then overall, if you see this, and these are the. So we generated for almost all several thousand of genes, and we have picked up only top 20 genes which are available in this figure, last page. And these are the microRNA which is binding. So that shows that our method is working, and these genes have indeed have a role in cancer, and their microRNA also is known. So the LET7B has a role in these genes, which is found here, and it's a way pre-validated. So basically, before we did, there were experiments which proved it actually. So that shows that they deep learning, again, machine learning and deep learning both has application. We need to train, and when you say that we did this, we do not do one time. We have to go back forth and do it again and again and again and again to achieve, optimize it. And so this the microRNA and deep learning method is available on our GitHub page. So if you want to practice, you go there. You can get the codes, examples. And I'm coming to another good topic, that image analysis, how image analysis is performed in deep learning. So I call it image deep. So recently there is a paper which predicts deep learning model to predict microRNA expression with the expression pattern matching with the tumor image slides. So basically, what it says that what it does that is take the tool is called He2RNA neural network. They take TCG RNA-seq data, and then they take the image-based figure whole slides of image of different tumors. Okay, and then they look if the tumor color or tumor is looking like this, whether the corresponding gene they can identify based on the colors, identify the expression pattern in the transcriptomic data. And while doing that, they use obviously the hematoxylin and eosin based staining slides, and they have created themselves also, and this is available from Okin lab. And then they from image itself, can you correlate from image itself that what is happening in the cancer gene and their transcription or at RNA, what expression pattern at RNA-seq level. So that's what we are doing, and we are processing, and we are looking for people who can actually work with us. With this, I will end here. But what we are going to do is that we are going to practice so these are theoretical aspects. So if you want to start, how you can start? That's a very valid question, right? And so maybe I can take theoretically also, and I can take some practical session also on this, that how one can do, how a person who want to start, how the person can start. So one of the first thing that one needs to learn Python and R. So then you can say, okay, I'm not good in programming. And no, you're not. It's not a question of being good or bad in programming. You need to practice, and then when you practice, you will be able to do it. If you're not practice, therefore you see yourself into programming is nothing but cook. If you're cooking, you're doing a programming. If you understand. So you know how, if you want to cook rice, what you have to do. You need rice. You need to wash the rice. You need to soak the rice in water, boil it, or put it in a pressure cooker. So same way, if you can chunk your task into small pieces, you can do a lot of programming. And so programming start with, so if you're not aware with programming, and many of you are from technical university, so probably you know it, and I'm sure you know it. But what I'm saying that one person who is not knowing should be inspired and start doing it, and anybody can do. So a lot of people who they don't have formal training even, so they don't even have degree, and they do good Python programming, because just that you have to understand the concept. So today what we are going to do, that we take this concept in for you a bit more, and let me see. In this, so first thing first, you need to install Python. Okay, so anybody who has seen the Python, I'm running a Windows terminal, so let me exit from here. Control C, Control D. Okay, so I'm in the Linux terminal, and if you are not aware of Linux, then you have to. It's another thing. So, but you can install Linux now in Windows. So actually, I'm using in Windows. and then then I have Python 3. So I enter in Python 3. So let me take to some example which we have used. Okay, suppose I have a word called bioinformatics. Yeah. Now I want to check only the first one of the word out of it. So how I am doing it actually? I'm getting it. Oh, actually, so I have restarted it. So now this bioinformatics is there. So let me have to run it. Let me go to the history. Okay. So okay, so the bioinformatics word is broken into pieces when I said list into bioinformatics. So now I just want to provoke you that you go and learn. So how the screen can print this. So if you learn Python, you can do it. So maybe I have some more code open for you. I'm just telling you to learn Python. And then in 1 hour, I'm not going to teach you. But what I'm going to do is that I'm going to, okay, run Anaconda. So you need to download Anaconda. And then Anaconda has Jupyter Notebook and Python in a terminal. Okay. So let me run the. So in your computer, you have to check whether you have Python, you have Anaconda, and you have to start practicing from half an hour daily. And some of the few examples I can give you for start programming. So because this training is not trained for start how you will start programming. But if you don't know, some of you can make a group and we can discuss with some of our team members, or some of our videos are online. So you can ask, or you go to yeah. So why not to go to the video actually? So everybody is watching YouTube. Everybody has a Geo. So let's go to YouTube. YouTube. So you can get a lot of videos actually, but if you just want to train from computational, in some minute we'll start actual analysis, computational genome biology. So you just look for computational genome biology. Let me see. Okay, so yeah, I have the message also here. So we are running a channel here. On this channel, you'll find a lot of information, a video from different talks, and there I have already explained introduction to programming recently, and then you have also a Python programming tutorial. Okay, so those who want to those who want to use it can go and do it. And still, if you have question, we can actually answer. So one need to sit in computer and do little bit every day, and then or also you can go to an organized course. But what I normally prefer that even if you are not having time to go to an organized course by the university, you just can train yourself by using any of the method online tutorial from python.org, or following some YouTube videos, or following your peers asking others. So we are going to start Jupyter. So so let me, if it is not working, वीडियो चल रहा है, ऑफिस चल रहा है.
So we can do in Google Colab.
Okay, yes. We can do it in Google Colab also. Yes, Google Colab is another way. Let me introduce you to Google Colab. So we can go to the Google Colab. Google Colab, if you have any problem. So what is Google Colab? Google Colab is Google's platform for doing machine learning. Google Colab. Colab. Okay, so Google Colab is providing us a platform for running uh, programs. You can run a program in Python, and it gives you uh, a resource for running the programs, and that one we can do it. But let me restart the Anaconda, because we have the everything written in Anaconda now. So Anaconda will run on local platform, and similar way Google Colab is also working. Yeah, so this should start. Okay. Okay, so let me, you are able, yeah, so now it's started. Okay, 1 minute. So we go to Anaconda. So you can take Jupyter Lab or notebook. So it will start you kind of this. And what is the advantage of this terminal? That you can keep your notebooks there. So some notebook we have, and then suppose you want to create a new notebook. Once you have it, so you have a Python 3 notebook. So okay, and then you can name this test notebook like, say, course. And then if I just want to say something, so there are in Jupyter, there are code, markdown, and raw, and heading. Suppose I want to give this as a heading, so I can go here and I can say course, and I run it. So what happens is become like a note. So it will stay as an intact note as long as you keep it and do not modify it. And then we can go, I have a test note here. So I just want to say hello world. So I just copying this one to this one, and I wrote it. So it's printing. So print is a Python or any programming language you start. The hello world is the first command. So print is printing the command. Suppose you want to write 2 plus 2 plus. So I have it in here. So 2 plus 6, and then run it. What will happen? So it will work as a like a Python interpreter, and it will add you, and you everything you do here is a store. So that is the, you can save it, and then it is stored. So if we close it, suppose I just close it now, so it will be here in my, it will be stored in my default folder wherever it is. So let me see, last modified. It should be here only. So let's see in my, okay, last modified. Yeah, so this one is here and I just need to open it and it open up. Yeah, so if I run it again, I can again see the next, whatever we do, it will be stored. And this can be taken, you can take mine, I can take yours, you can put it in Google Colab, and everywhere you can add it and you can use it actually. So wherever you can put it, you can use it, or you can share with share with everyone. So that's the advantage of using Jupyter. And so what we are going to do today, that we are going to use is use one of the existing code which is available here. Let me see. Okay, so and then you can take other advantage of this program is that you can take from somebody else. Yeah, so you can take from anyone from who you like, and you can take a lot of things. So I am taking one COVID-19 projects which we have run. So what this is doing here is that it is installing libraries from the we have already installed actually. So if you don't know how to install a library, you just suppose it's a Numpy, you just I can do it here also. I just need to bring this up. Okay, let me say there, pip3 install. Pip is the Python repository. Python install Numpy. So if I'm doing it, let me run it first. So it's actually already there. So I know, let me use Python 3. Should run at least and show. Okay, so it actually knows that it is there. So therefore it ran over and said, okay, it is there. So if you do not have in Jupyter, you can install in Jupyter. Suppose I don't have, sorry. Suppose I do not have here. Maybe I just want to check whether I do not have here. Control D. I just say pip3, then it says pip itself is not found. Okay, here in this Python interpreter, pip3 will work actually. Okay, so if the Numpy is not there in my computer, it will install. So now it's installing. So you said, okay, since you told me, I know which Python library to install, but how can I install a Python library of my interest without asking somebody? Yes, so that one can do. So you need to go to pip and pip Python. You will get a library. Yeah, you will get a page. This is called this in Python packages library. This website. Suppose you are looking for genomics. Yeah. So you can look for your keyword, whichever area you are working in, finance or whatever. So it will give you the entire keywords, different libraries which are available, and then one need to click. Suppose Keras Genomics you want to install. So this will give you a hint how to install it. Okay, so you just need to copy this, and once you know some of it, you will start doing it. Pip install. If there is a spelling spelling mistake, it will give you an error, and then you will know, okay, I have to correct it. So it's a very big library, and if you look at this many projects are there. So you can browse projects and you'll look into, and if you develop something, you can also pull it, because you can log in by registering. You can pull it out with your friends and with whoever. New library you are generating, you can also add to it. Okay, so with this we go to the again back to the COVID-19. So we have already installed different libraries, and this code will be available to you for using it. So what we are going to do is run this code first. So this line is not required. We have already have tested that it is there. So I'm just taking it out. So whole idea is that one can modify, change, read, and one can also add what is it, what am I doing here? I'm installing different things, so I can edit it and do it in my way, and make a note also. So let me run this one, and this is running. Now what I'm doing is that I'm taking confirm data, and data is available on somewhere in COVID-19 data set. Okay, and data is actually actual COVID-19 data set. It's changing every now and then. Okay, so you see the data is available, changing dates and all are available. Country wise data is available actually. So we will run this, and then we'll run this one again and to check it. So we are seeing that data from Afghanistan, Albania, or n number of countries are available here. Then what we want to do is that we want to do some computing to the data we have received, and then we just run it over. So what we have done here is that on each day, what is the percentage, what is day by day, what is the death rate. So as you see, the year started with 0 death, then when you go to somewhere in June, July, it's July, the death rates are different, and so on. When you go to different data points, values are different. Okay, so now what I want to say to confirm dates, world cases, total death, mortality rate, recovery rate, total recovery, total active. So what you see the on the dashboard of different countries, actually it is generated from this kind of code, and they have wind up. So this is available here. The array has world cases and world cases with a country-wide profile. So it's there, and now we go to the next one. We will see the country and all in short while, and then we have daily increase and all are compared with the range of Windows 7 days in the 7 days profile, and all these values you will see in some minute. So then we run it over world cases 50 to 55. We see some data. We have days for 10 days. The data so we are checking the data, and then you will see shortly some graphs and all. And so the code will be available. You can try this code, and we'll run it over period. So two students who were training, they actually generated took the idea and run the test versus as some support vector machine is used here to see graphs, and then we keep on going. You will start seeing the country-wise distribution. So in last 7 days, what is happening of the coronavirus cases, and with respect to different dates. With respect to date on 1st of January 2020, and then we go on, keep on going, and then we see that confirmed cases in India, different countries we have done, and then there can be some error because the program is developing on the other terminal, but it will run. Some of them will run, and then whatever is not running will debug it. One can debug it over it, or maybe a package is missing. So now we just run this, and we see that this is making a different regression graph, and every graph is different. At the end, we will see a pattern. Okay, so on this date, total confirmed cases worldwide is this much, and every day there is some increase. So you see a pattern of graphs, and this is as if today. Yeah. So when you run today's, And if I run it 5 days later, it will be 5 days later. So yeah, so this is running, and we have this one running. Then we go to the next one. So if the last one is not finished, the next one will have a problem. And then we see what we have seen here, that we have divided this into country wise, confirmed cases versus death, recovery, blah blah blah, India, USA, India, Brazil, all the countries. Yeah. So maybe we can run it once again, and now you see this graph for Indian states. Okay, so what is the status in Indian states? So all this is organized, placed. An advantage of the Python node is that if we put this in GitHub, or we put it any source, or give you can edit and run yourself. So we have this confirmed cases in different states. So the graph is done. So we can choose which state we want. Pie chart we can generate, and some of the functions are not working. We will debug it why it's not running. So since we have put it, means it should work. Maybe a package is missing in this Jupyter. Maybe we have used this project is live project run by 2 trainee students who trained recently with us, and they discuss with that, well, this is what we are doing, this is the code we have written. We will check every now and then. And so you see that this is one type of I'm giving you an example. This is one type of data which is available and we are using. The data has to be available. If you have learned some Python and Jupyter, running Jupyter, you can take any other project and start doing it, right? We are not restricted by time and restricted by anybody's help. You can do it at home, because this Jupyter, you saw that my computer, you don't know how fast or slow my computer is. It requires only a normal notebook to run. And if there is a problem, you can also, one of you told that one should go and Google Colab and use it. And but since I had it here, I just do it. So basically you can bring it to Google Colab, and then I save it now and then close it, and then I give it to you, you can use it. Okay, so this is one example, and there are thousands of such projects are running and available globally that you can collect, you can gather, and you can modify, optimize for your requirement and start using it. So that's what is our that's what you need to do it, and then you will use different machine learning methods, more and more machine learning methods. And suppose you want to start, first thing is to learn Python, and probably many of you know, and then start practicing. So you can say, okay, I'm not knowing how to practice. You can go to database called Kaggle. Your machine, so you can create a login there, and you can get a lot of projects. And even if I do not have projects, so I just want to say genomics here. Suppose we are biologists, and we done. So there are a lot of projects are running here, and data sets are available, notebooks are available. I've just said genomics, and then I get genomics notebook. I say proteomics genomics notebook. So you can start reading the notebooks, what they have done. Sometimes it may be useful, sometimes it may be not useful. So that this exploration data for library function definition. So there's using for generating graph. Okay, they may use some type of graph they have generated. Initial impression, my initial impression is that, because I have not, I probably never seen this graph, yeah? This project. So you get an idea immediately, and now this your requirement? Is similar is your requirement? Will you want to try this and then use on your own data, or no? So and then you have different projects, but then you also have projects on in different topics: earth, art, health, computer, data cleaning, data deep learning. You just let's take a project of health. There are different data you see, and they are also running competition, so you can solve some of their competition. But what I am going what one can do, that you need to see the data. You have to follow some of the data, understand how people have done, and these are very they're only limited. So if you carefully look into if you look carefully into the data sets, what you see that uh, there are only 10-12 packages which are used primarily in all data sets, or there are 15, 14, 16 will be another one which will be used by another software. That will be a specialized requirement, and you can you can do that a specialized one. So one which we have used here are the most common ones are Numpy, Matplotlib, Pandas, obviously random time. These are general ones, but Pandas, Numpy, Matplotlib are the major. Scikit-learn are the major. That only 20 tools or so which you need to learn, and which will be and these are employed. These are just called, and then you are using reading the data. So the pandas is reading the data as a data frame. Yeah. So from some source, source can be from your papa's computer, force can be GitHub and a web host, or your institute website. Whatever you want to give, you can read it from location and permission you have. You can read it, which you are already knowing, right? So that how to fetch a data from internet. So one can fetch the data from internet, and then this is one thing. So and then what else one can do is that you need to prepare yourself with respect to the data set. So maybe I just give you a short, not full, but the short introduction about packages, machine learning. So I'm not sure where is it, but I can give you a yeah, maybe this one. Okay, so these are major packages. Let me have okay, so these are this one is just installation. So second one maybe okay, so as I told you that you need, what kind of packages you need is obviously you need Python, Pandas, Numpy, this Seaborn and Scikit-learn. And how you are installing, you can install it and then use as an import. And these are different modules. So you have, if you want to install, suppose you never install it, you can install one by one, or you can install at once. And then it's installing. See, this is installing, right? And then you can do whatever computation you want to do, is data structure. So that you need to train yourself a little bit. And then since we are talking about, I think this should be, there has another slide I have actually. No, this slide is not available here. But what I wanted to tell you, that you need to know if you know 20 packages and they start using it, 5 more packages you will need after 1 year. So keep on practicing, and then keep on using it. Keep on using for a small tasks. Say COVID-19 prediction. There are a lot of people are working, so you can take their example, take our what we have done, and check it how you can improve. What kind of improvement you can ask yourself some research question. That okay, I want to see the mortality rate in India in September, July to September, and what happened suddenly the cases are very less in November, December. Is virus is there, or what happened actually? Why is the so less numbers? Is that sampling is still going on, or that there is an end of pandemic? So a pandemic is, many people believe that in India, pandemic is approaching towards that end. And so is it really a dead end? So from data, can you make such kind of a correlation? Apply to data. You apply uh, different statistical package you can do, and with data you can correlate whether what is going on, and make a lot of statistical prediction, and which you probably see, say, in the print.in or other news channels, and say New York Times. So they all have the data scientists who are sitting in the background and doing such analysis. So and they are using same maybe Jupyter only, and we are seeing it. Since time is still there a little bit. So one package which if you are not familiar with Python and you don't have time, one package which I think you should know is Orange, where you can use Orange package, Orange ML. Okay, so Orange for data mining. Okay, so the Orange, because it's orange in color. So Orange you should install. It's easy, and I can show you, discuss with a screenshot. So you can keep your data, and then data has the values and all. You can see that. You can see your data, what kind of data it is. You can create the classes. You can just show, if you need to train, there are a lot of YouTube video for this, and then once you know it, you can start using it. So for biology, it is very extensively used. So this is kind of iris data, and data classifier is used here. So one can use, but still you need some days of training. And then in this package, what is there, that if there is a file option, you can open file using this. Then you can connect to different type. So you can actually, to file this, you can connect to different type of units, and it's a kind of a modular programming. So basically, when I say file, there is a two file you want to concatenate. There is a concatenate function. You can just connect to it, and here you will see concatenated. So these are two files, right? So 1 and 2, and open. Then they have concatenated this file, and then you have a data as a text, data text. So basically, you get the results. So they have made it in a way that it's a virtual visual programming. So anybody can use, and obviously you need to train a little bit for yourself. And they also you can use any machine learning data, or any data set which you want to use. You can do different tasks in this. So in COVID-19 data also can be used. The trainees were using it, so you can also use this. And then you can draw a heat map. There are a lot of things which are inbuilt here. And then you see that this classification algorithms can be used. Then you can use for viewing image. So image analysis can also be done here. Then you can also do clustering. Hierarchical clustering is done here. Then you can do scattered plotting, and all other type of experiments you can do. And this is primarily made for biologists, people coming from biology background. biotechnology background, and other subjects. And yeah, this is a logistic regression and random forest ROC curve is drawn. So is all so these algorithms are inbuilt here as a boxes, and you can use the boxes. ROC curve is also available as a boxes if you go here, and then you can use it, and it's very easy in a way that you can, yes, you see I'm just showing you the examples which is the pre-existing existing with them. So and you one can use, so you need to take their own example, run it, and you can still visualize this still going on. So many examples are pre-existing in their screenshot option, and then you can start getting data distribution and other beautifully described way. You see the different graphs and the different, you can also generate confusion matrix, which is here, and you can generate evaluation result, you can generate your word cloud. And word cloud means you have several words and you want to see what is the occurrence of the word. So you see that a lot of things you can do here. And this is from developed by a bioinformatics group actually. So it's bio focused. So but it can be used in beyond biology also. So you see the lot of information. So lot of so in rather than looking for MATLAB or other algorithms which are commercially available, you can use, there are a lot of other tools which you can use if you are if we immediately has to do before learning Python properly, you have to do the analysis. Obviously, I recommend learning Python, and I like to recommend learning these tools as well. So there is another tool called K9 or 9. I'm not sure if you're aware. This is also doing good job. So data K9 innovation. So yeah, so as you see the graphs, you can see that. So they're slightly different concept, but these are also, but both of these are freely available, and then you can use it for creating your data science project. And those who are, as I told you before, also those who are interested, please let me know that you can, if you want to do it, and we can do it. So what I will do here, that I will send you a link of our COVID-19 data that you can practice later on your computer, and practice it. You will get it from you. I will send it to Minaji, the GitHub link, and then you can download it and play with it. Play with other data. Okay, so I'm open for